Hi guys, welcome to my presentation on Chapter 22, Evolution by Natural Selection. So first, I'll talk briefly about how people thought about the different species before the theory of evolution. So people used to think that species are unchanging, they don't change over time, they always look the same, and that they're unrelated, so that they have no relationship to each other. People used to think that they were created independently from each other. And they also used to think that some species are higher and some lower. So, some species are better than others. So, Plato thought that species do not change over time and they're unrelated to each other. So he thought there's just different types of living things that have no relationship to each other and will always look that way. When Aristotle came along, he updated that way of seeing the world to include the scale so that, people, of course, are on the very tippy top of the scale, and then it goes down from there to your simpler animals and then to the plants on the very bottom of the scale. So he thought that these plants are inferior to people at the top of the scale, and that a worm is inferior to a fish, and so on and so forth. And that's basically what we thought about species until the theory of evolution was proposed in 1858 by Charles Darwin and Alfred Russel Wallace. These are two scientists who came, the idea of evolution came to them independently, so they were working on opposite sides of the world and they really didn't influence each other. Charles Darwin had been working on the theory of evolution for a long time, actually, but he was nervous to publish about it because he knew that people weren't going to like it. So he had been working on his book on the origin of species, in which he presents all of the evidence that he had gathered for evolution, but he was nervous to publish it. And then in 1858, Alfred Russel Wallace sent him a letter just sharing Alfred's own idea about evolution with Charles Darwin and asking basically for his advice. And so Charles Darwin wrote back, yeah, you should publish this. And to himself, he thought, well, this means I need to publish my book now. And so they ended up both publishing in the same year. Charles Darwin is the scientist who gets credited with the idea of evolution, because whereas Alfred Russel Wallace just published this paper where he presented the idea, he didn't have any evidence. Charles Darwin wrote a whole book which he had a lot of evidence in, so he got the credit. The theory of evolution basically states that species change over time and they are related to each other by common descent, and that natural selection is the process which has driven this. So species change over time, so they may not look the same way. As time passes, they'll start to look different. Individuals do not change like that, but species do. Whole populations can change. And eventually the change can become so great that you actually have a new species created, and that's called speciation. So speciation is the creation of a new species by evolution. As long as the new species does not breed with the old species, it will be considered a new species. This process has created all of the species on Earth. So that means that all species are related to each other by common descent. We all share ancestors with each other. And then finally, natural selection is the process that has caused this to occur. Natural selection is just the idea that you have individuals of a species in a population have different traits. They don't all look the same. They have differences between them. All those traits are heritable, so they can be passed on from parents to offspring. And some of those traits are advantageous and some of them are disadvantageous in the species environment. And then if you have those three conditions, variable traits, heritable traits, advantageous or disadvantageous traits, it's inevitable that individuals who have the advantageous traits are more likely to have children. Individuals who have disadvantageous traits are less likely to have children. So then the next generation of individuals in that population will have more of the advantageous traits and less of the disadvantageous traits. And this process will continue until the whole population has changed. It's frequently driven by limited resources, not all the time, but frequently there's not enough resources for every individual that's born in a generation to survive. And so the ones that do survive they're more likely to have the advantageous traits. Individuals with disadvantageous traits are less likely to be able to get enough resources and are more likely to die. So in this example, here we have moths on a tree. So if you have this population of moths, some of them are dark and some of them are light, and if they're living in a forest where the tree trunks are dark and they like to hang out on the tree trunks, the dark moths have an advantage there because it's hard for birds to see them, but the light moths stick out really well, and so if a bird is coming along looking for a moth to eat, it's going to see this light-colored moth right away and eat it. It's not going to see the dark-colored moth so easily, so most of these dark-colored moths will survive and not get eaten. So being dark-colored is advantageous in this environment, and as time passes, more and more dark-colored moths will be born and have children, whereas the light-colored moths will have a hard time having children because they're being eaten too much. So eventually, you might even see all of the white-colored moths disappear from that population. So that is evolution by natural selection. On the other hand, if you took this population and transplanted it to another forest where you had trees that have a light-colored bark, then the situation is completely different. Now all of the light-colored moths blend in really well, and the dark-colored moths do not. They stick out. So now the birds are more likely to eat the dark-colored moths because they can see them better. And the light-colored moths are more likely to have children, they're more likely to avoid being eaten and have children. The dark-colored moths are more likely to be eaten and not have children. So over time, moths in this environment will evolve to be more light-colored and have fewer dark colors. So what is an advantageous or disadvantageous trait really just depends on the environment that the population is in, and it changes over time, and that's really what drives the change in species over time. Different things become advantageous or disadvantageous as time passes and as populations migrate to new locations. So we'll talk about some of the things that inspired Darwin or that gave him the idea for evolution. So first, he took a lot of inspiration from his journey through the Galapagos, where he was basically going as a naturalist to study all of the life forms that were there. And the Galapagos are a bunch of islands off the coast of South America. So here you have kind of the general map. This is South America, and these are the Galapagos Islands here. Here they are again and again. This is actually showing the journey of Darwin's ship, the HMS Beagle, through the Galapagos Islands. So he journeyed there for several years and studied the finch, different species, but he became interested in finches. He noticed that there's many different finches on these islands, and so he categorized them all into different species. He noticed all the different species that were there, and he was studying their similarities and differences. And so it just seemed really strange to him that you have all these different finch species there, and they all look very similar to each other, but they're all different. And he would go to one island and find one type of finch there, then go to a different island and find a different type of finch there, and it was just very confusing. But of course it makes sense if you figure that all these finches evolved from a common ancestor who was a finch living in South America in the forest here. And if you just had a couple of individuals of that finch species make it all the way out to the Galapagos Islands, then they would start evolving independently. If they went to different islands, they would evolve independently from each other to create different finches. He was also inspired by his hobby of pigeon breeding. He liked to breed pigeons. Pigeon breeds can look really crazy and pretty cool. So over here you have wild pigeons. This is basically the two kind of ways that wild pigeons will generally look. Sometimes you'll see like an albino or something, but most wild pigeons look like one of these two guys. But pigeon breeds can look all types of ways, and as you notice, these birds some of them look very crazy. Like, what the heck's going on there? They all look extremely different from the wild type pigeons. But they all are pigeons, and they came from wild pigeons. So people would catch pigeons and breed them together, and eventually you get all these crazy looking pigeon breeds. And Charles Darwin liked to do that himself. So this really underlined for him that it's obviously possible that species can change. If it wasn't possible for a species to change, it would be impossible to get all these crazy looking pigeon breeds by breeding wild pigeons. He was also influenced by his friend Charles Lyell, who was a geologist, a very famous geologist actually. Charles Lyell is the first person who, I guess, published a book that convinced people that the Earth is actually very old and that it has changed over time. So people used to think that if there's, say, a canyon in a particular place, then there's always been that canyon there. It has lasted forever, well, as long as the Earth has lasted. And they thought the Earth was, around 6,000 years old, at least in Europe. Charles Lyell basically demonstrated that actually that's not the case, and all of the geological processes that we see happening now, like wind blowing and water flowing, have happened throughout time and have very, very slowly created all of these formations that we see. So if there's a canyon in a place it didn't used to be there millions and millions of years ago, it might not have been there, but it was carved out by water and wind. So that kind of demonstrates that the Earth must be extremely old for these very slow processes to have created all of the massive geologic formations that we can see. And he also had the idea that if you're looking at like a canyon or a cliffside or something where you can see different layers of rock, those layers are in order of their age, so that the topmost layers are the youngest, and then as you go down, you have older and older and older layers of rock, and the bottom layers are the oldest layers. They've all been laid down in order. So Darwin gave him the idea that the Earth is actually extremely old enough that it's plausible that species can have changed a lot, so much to have created, very much new species that look nothing like the old species. And there's another guy, William Smith, who's also a geologist who influenced Charles Darwin. William Smith proposed the idea that fossils, the age of fossils, can be estimated from the rock layer that they're found in. So if you find some fossils up here in the top layer, those are young fossils. If you find some other fossils in the middle layer or in the bottom layer, those fossils are older, and the bottom layer fossils are the oldest fossils of all. So he actually created this map. It has the different rock layers here, organized by their age and by their depth. And then next to it, he has the different fossils that you can find in those different layers of rock, where he was working, which is in England. And so he used this kind of map to organize these fossils throughout time. So these would be the oldest fossils down here, and then you get some slightly younger ones up here, and they get younger and younger and younger as you go up, so you can actually track the change of organisms over time. Which of course indicated to Charles Darwin that organisms change over time, or not individual organisms, but populations. He was also influenced by the economist Thomas Malthus. Thomas Malthus proposed that populations are limited by resources, so we only have, X ability to produce food. The food that we are able to produce can only feed Y number of people. If you have more people than that, then people will starve because there's not enough food. So he proposed that this kind of limits population naturally. So if you have a graph where this is the production of food, it grows slowly over time, but the population can grow much, much faster. It can actually grow exponentially, theoretically. But once the population gets bigger than the ability of the food supply to feed that population, then you would have a Malthusian catastrophe, which is basically where many people starve. So Thomas Malthus's ideas were very popular for a long time, and they do make sense. But there's one thing that he didn't take into account, which is technology. That technology can produce dramatic increases in our ability to produce food. So, for instance, this guy Norman Borlaug. He was an agronomist. He bred wheat, and he was able to create a new strain of wheat that yields a lot of wheat. So you get a lot of calories out of it, but it's also very resistant to disease and to harsh climates. So it can grow in a lot of places very, very happily. So this new strain of wheat has been grown all over the whole world now, and it really hugely increased our ability to produce food. And it's estimated that about 1 billion people have been fed using the wheat that Norman Borlaug made that could not have been fed otherwise, so that he would have saved about 1 billion people from starving to death. So if you take into account technology, then the graph looks very different, where you can have your gradual growth in food production and then a sudden sharp shoot up of the food production if you have a new technology introduced, like Norman Borlaug's wheat, so that theoretically you could keep up with population growth if you get enough new technologies fast enough. But Thomas Malthus's ideas about populations limited by limited food resources were very influential for Charles Darwin, and they gave him the idea that if you have a bunch of organisms born in a new generation, not all of them will be able to find enough food to eat, maybe, and so some of them will die. And then he thought, well, the ones that are less good at surviving in the environment are more likely to die. So that kind of gave him part of the idea for natural selection. So now we'll talk about some misconceptions, some common misconceptions that people have about evolution. First, by far the biggest misconception that people have is that evolution applies to individuals, so that, say, an individual wolf could turn into an individual dog. That's not how it works. Or an individual monkey could turn into an individual human. That's not how it works at all. Individuals do not change like that. Individuals look the way they look for their entire life, and then they die. But they also have children, and their children are genetically different from them. So the population over time can change as children look different from their parents. And you have some children surviving better than other children, some parents having more children than other children, or than other parents. So populations change over time through evolution. Individuals never do. Related to that, you have kind of the difference between acclimatization and adaptation. In adaptation, you actually have mutations that make you better suited to living in the environment where you are. So someone could be born with an adaptation to high altitude, and then that person would have that for their whole life, and then their children would have it as well. So for instance, people who do live at high altitude, like the Sherpas in the Himalayas, they are adapted to living in that high altitude. They actually have a different form of hemoglobin in their blood that is better at carrying oxygen. So they're better at getting oxygen out of the air than people who live at a low altitude. They're adapted to that high altitude. If you as an individual go up to a high altitude, you will not adapt to it. You will acclimate to it. So if you're kind of some white dude who wants to go walking on a mountain, if you go kind of slow, if you increase the elevation that you're at slowly, then your kidneys will start making more of this hormone EPO, erythropoietin. That is the hormone that tells your bone marrow to start making more red blood cells. So as you go up in altitude, your body is going to sense that there's not enough oxygen for you anymore, and the kidneys will respond to that by making EPO, which will tell your bone marrow to make more red blood cells, and then you're going to have more red blood cells, and that means that you can carry more oxygen. That is not an adaptation. That's not a mutation. That's just your body adjusting its homeostasis to the new environment. So that's acclimatization, and it's different. So acclimatization occurs in individuals, adaptation occurs in populations. Another misconception is that evolution has a goal, or that it's headed towards, a perfect organism. This is not true. Evolution is not goal-directed at all. It's completely random. It operates by random chance. It's random chance that gives you mutations. So what mutations you're going to get is entirely dependent on chance. You may get one that, helps you adapt to a new environment, and you may not. And you're not necessarily going to get the one that helps you adapt the best to that environment. So there is really no goal in evolution at all. And that also means that there's no hierarchy. So there's no such thing as higher organisms and lower organisms. We're all equal. There's also no perfection in evolution. Evolution does not create the best organism. It just creates an organism that is good enough to have children in its environment. That's all. Particularly any problems that affect you after you've had children, it's impossible for evolution to select against those so that their incidence will be reduced in the population. So for instance, some people have a genetic predisposition towards diabetes, but that doesn't prevent them from having children, so it's not selected against. They actually don't have a disadvantage from an evolutionary perspective because they have an increased chance of getting diabetes. Getting diabetes does not prevent you from having children, so it's not disadvantageous evolutionarily. That means that evolution will never eliminate a disease like diabetes from the population, or a disease like cancer. Anything that strikes after you've had your children, it's impossible for evolution to eliminate it, or even to reduce its incidence. And then finally, the last misconception that was discussed in the textbook is that it's not just natural selection that drives the change of species through time. Natural selection is the main one that we hear about, but there's also other types of selection. For instance, sexual selection. That's when individuals choose a mate based on some characteristic that they have. That would be the explanation for why a bird like a peacock looks the way it does. So peacocks have a really long, pretty tail. That's not good for them from a survival perspective. It makes them very obvious to predators, and it makes it harder for them to run away from predators. So it makes them more likely to be eaten. But female peacocks really like the big, pretty tail, and they're more likely to pick a mate who has the biggest and prettiest tail. And that is why they have, such a pretty tail. So that is sexual selection. You also have artificial selection. That does not occur in nature, but that's where people take organisms and breed them together intentionally to create certain traits or to eliminate certain traits that are not desirable. So the pigeon breeding, for instance, that's artificial selection. Dog breeding is artificial selection. All the breeds of pets and domesticated animals that we have been created by artificial selection. And then finally, you have genetic drift. You actually have others, more than just these three, but these are kind of the main ones. So you have genetic drift or gene drift, and that's basically random changes in the genetic makeup of a population. It's not driven by anything other than just random chance. So this is an illustration of gene drift. If you start with this population of beetles, some of them are brown, some of them are green. If we figure that there's no advantage or disadvantage to the color of the beetle, then there's no way for this population to evolve by natural selection, at least not in terms of what color the beetles are. But you could have someone come walking along and not even be looking down and step on some beetles. By random chance, this person has stepped on only green beetles, and there were only 3 green beetles, so now there's only 1 left. So now the makeup of the population is different. There's more brown beetles, relatively speaking, and fewer green beetles. If this happens again, if someone else just randomly steps on a green beetle, all of the green beetles will be gone, and that's just due to random chance, not due to any disadvantage associated with being green. Finally, whoops. Okay, I accidentally pressed the button to go forward, and now I can't go back without erasing what I said, and I'm not trying to do that. So when I was in high school, my physics teacher obviously didn't believe in evolution because he had a poster about how silly it was on his wall in the classroom, and the poster said that if you believe that a princess can turn a frog into a prince with a kiss, that is a fairy tale. But if you believe that lots of time can turn a frog into a prince, that's evolution. This is a misconception about evolution. Evolution is not suggesting that just time makes species change, and it's also not suggesting that an individual frog has turned into a prince. More just that the population of frogs has evolved over the vast stretches of time, not because of the time, but because of natural selection, because not all of the frogs could survive in every generation. Some of them died, and ones who had disadvantageous traits were more likely to die, and so the population changed over time, and eventually, after many years, with that natural selection occurring, then you get speciation and new species out of the frogs that don't look like frogs anymore. But without the natural selection, you wouldn't have that. So if you just had a lot of time pass, but no natural selection at all, no advantageous or disadvantageous traits, then the frogs would look the same as they always have. They would not have evolved, unless of course you had some genetic drift, just random chance, but there wouldn't have been really evolution. So now we'll go through some of the evidence for evolution. First off, time is big. The Earth is very, very old. We used to think that it's not very old, but now we know that it's incredibly old. And we've learned that by studying rocks. So Charles Lyell studied sedimentary rocks and found out that in order for them to have formed, the massive formations that they can form, the Earth must be extremely old. So you can actually look at, say, sandstone, and you can measure the rate at which that forms. And it's extremely, extremely slow. And then if you look at some of the giant sculptures of sandstone that there are in the world, natural sculptures, of course, then you can do math to calculate what the age of the Earth would be, and it's very big. It's very old. So those early geologists created the geologic timescale, which is massive, and based on the layers of rocks that you find fossils in, you can put the fossils, you can relate them to that geologic timescale. So down here, fossils from this really old layer of rock, those fossils are about 510 million years old, based on the layer of rock that they're in. The ones from the top layers of rock are about 275 million years old in this particular location, which is the Grand Canyon, actually. So that was kind of the first way that we started dating fossils to find out how old, not only how old the Earth is, but how old life is. But now we have better techniques than that. We have radiometric dating, in which you're basically using radiation to measure the age of something. So if you have a tiny bit of a radioactive isotope in something, which you always do, that decays to something else on a very reliable time scale, like you could set your clock by it. And so if you know how much you have now in the sample and how much you should have if none had decayed, then you can know how old that sample must be. So based on that, we've been able to estimate the age of the Earth at 4.6 billion years and the age of life at 3.4 to 3.8 billion years. So that's certainly enough time for some evolution to have occurred, even if it's happening very, very slow. We also have the existence of extinction. Extinction is a type of change in species over time. So they used to think that there's no extinction. Like no scientist took that seriously. It was considered wild speculation that an entire species could go extinct and stop existing. So we were finding fossils of organisms that didn't look like organisms that we have today, but scientists were just figuring, oh, we haven't found it yet. It must be alive somewhere. We just haven't found it because we haven't explored everywhere. With this guy, Baron Georges Cuvier, proved to the satisfaction of other scientists in 1812 that there is extinction, and the way he did that is by focusing on fossils of very large animals. So for instance, this giant sloth here, it's several times taller than a man, as you see. So we have fossils of this animal, but obviously we don't see any of this animal around. So he found several examples of animals that are really, really huge like that in fossils, and he just pretty much said, we've been all over the place, we've been all over everywhere, and we don't see no giant sloths, so they're not there. Like, you would see something that big if it was there, and they're not there. And that finally, I guess, made people realize that, I guess extinction is real after all. And if extinction is real, then species can't be changeless because it's a form of change. Actually, now we know that about 99% of all species that have ever lived on Earth are extinct. It's a very, very tiny minority of species that are still alive today. Next, we have vestigial traits. A vestigial trait is basically it's something that you have from a kind of a previous ancestor that you don't use anymore, and so it's gotten very small and has no function. So for instance, your tailbone. Everybody has a tailbone. We don't have tails, but the spinal column does extend a little bit below the pelvis. These actually start out as independent vertebrae, and then they fuse together to make the tailbone. That is left over from the tails that our monkey ancestors used to have. So monkeys and other, well, many primates have a big long tail that they use for jumping around and climbing in the trees for balance, and to grip. We don't have any need for that, so we don't have a big long tail anymore, but we do still have this little vestigial tail, the tailbone. It's evidence that we used to have a tail. Otherwise, why would we have it? You can see other vestigial traits in flightless birds. Every flightless bird still has wings, so penguins have wings, ostriches have wings, emus have wings. But they're not like big wings, like, well, you can hardly see it, but it's right here. There's the wing of that ostrich. It's not really, obviously, it's not big enough to carry that ostrich around in flight. It's not useful for flight. It's a vestigial wing, but it is evidence that their ancestors used to have wings that could fly. And then finally, we've got fishies, cave fish. Fish that have gotten trapped in water in a cave and have evolved there for many, many, many years. They don't have any need for eyesight because there's no light inside of the cave, so they've lost their eyes. They don't have any eyes. They're eyeless fish, but they still have the little eye socket. They still have the socket where the eye used to be, so you can see that they used to have an eye, even though they don't have one now because they don't need one. So, that little eye socket without an eye in it, it's like there used to be an eye there, you guys. And we have transitional forms also. So related to that, you have the law of succession, which is just something that scientists have noticed, which is that if you go to a particular area of the world, the species that are living there will be similar to fossils that are found there in some cases. So for instance, in South America and in parts of Texas, you have armadillos living today, and if you look in the fossil record, you can find this dude who looks very much like an armadillo, but not quite like an armadillo, similar to an armadillo. So it's like this is the armadillo ancestor, and you don't find this guy in places where you don't find the armadillo. Almost like there's some connection between them. Then here in Australia, you can find fossils like this, which look very similar to the wombat, which is alive today and is only found in Australia. So that's the law of succession. Then you have transitional features and transitional forms, which are basically just forms in between a modern species and an older species that you can find in the fossil record. So you can kind of track how features of that species, or of those different species, were changing through time. So for instance, this is an old fish, and the fin bones here, if you zoom in on them, they look like this. This fish is from around 385 million years ago, and here you have a more recent fish from 375 million years ago. This is Tiktaalik, which is a fish that was able to kind of push itself on the land using its fins. So you can see its fin is kind of more flippery. It doesn't have these long rays on it, but it has more of a meaty, flippery shape. And if you look at the bones, they're quite similar to the bones in the older fish, but they're also a little bit different. So you can track this through time. You can track these transitional fin bone features through time. And if you track it further through time, you come to this guy, who's actually more adapted for living on land. His fin is turned into a teensy weensy little leg, and the bones in it look a lot more leg-like and hand-like. If you go further in time, you get to this early reptile who actually has a proper leg and whose bones look very much more recognizable as hand bones. But these bones all correspond to each other. So you can see how that feature has changed through time, and that's the transitional form. Next, we have the fact that species that are related to each other are frequently clustered together geographically. So for instance, on the Galapagos Islands, you have a lot of different finch species, and it's kind of weird. Why is there so many frickin' finch species there? Well, it's almost like they have some sort of relationship to each other, you guys. There's all these different finch species on the Galapagos Islands. People are still studying the finches there and still finding new species. And it's estimated that around 1.5 million years ago, you had some finches from Ecuador make it over to the Galapagos. That would be one species of finch that came over to the Galapagos and then went to the different islands and started speciating on the different islands to form all these different species of finch. It would just be weird if there was no relationship between those different species of finch. It would just be pretty weird to find all of them there on those little islands. Next we have homology. Homology is different things that look similar to each other. We've got 3 different types of homology: genetic homology, developmental homology, and structural homology. Genetic homology is homology at the molecular genetic level. So you have the genetic code. The genetic code is the same for all organisms. So that's probably the biggest, most fundamental homology that exists. Every single organism on Earth uses the same genetic code. If there's no relationship between them, that is really, really weird. You also have orthologs. An ortholog is a version of the same gene in a different species. So for instance, you have a gene for your ribosome. Well, actually you have multiple genes for your ribosome, but for your ribosome you have some genes, and you would find orthologs of those genes in other creatures, in other species, all of them actually, since they all have ribosomes. You have a gene for the enzyme alcohol dehydrogenase. That is the enzyme that you use to metabolize alcohol once you've drunken it. Other creatures also have a gene for alcohol dehydrogenase. Plants have a gene for alcohol dehydrogenase, many of them. So that's an ortholog. The plant alcohol dehydrogenase gene is an ortholog of the human alcohol dehydrogenase gene. They're related to each other. They're not actually identical, but they're very, very similar. So if you look at organisms for which we have sequenced the entire genetic code, you can figure out how similar those organisms are to humans on a genetic level. And it turns out that our closest living relative, chimpanzees, share 98% of their genes with us. So 98% of the genes that you have in your cells can also be found in the cell of a chimp. 92% of the genes that you have in your cells can also be found in the cells of a mouse. You're 92% genetically the same as a mouse. Fruit flies, you're 44% genetically the same as a fruit fly. 44% of your genes are also found in a fruit fly. Even yeast. Yeast is a single-celled organism. It's a fungus. It's what makes your bread rise. It's also what gives you a yeast infection. 26% of your genes are found in yeast cells. You're 26% the same as a yeast. That's crazy. That's a remarkable level of homology, and it's just very, very difficult to imagine how that could have come about if there wasn't a relationship between you and yeast. Then we have developmental homology, which is similarities between structures that are present in development, which would usually mean structures present in the embryo. So embryos of mammals all have tails and gills. Here we've got 3 different embryos. This one's for a chicken, this one's for a human, and this one's for a kitty cat. Of course, chickens and humans and kitties do not look the same. They look very different from each other, but the embryos actually look really similar. All of them have a tail. The chicken tail is going to turn into something looking pretty different from that. Humans, of course, are going to lose the tail almost entirely, except for the little tailbone, and kitties have a tail. And all of them have these little gills. These aren't fully developed gills, of course. They're like proto-gills. And they're left over from when we were fishes. So as soon as we got gills, we never lost the gills. We still have them in embryo form, and then as we develop, they turn into something else that's not gills. And as soon as we got tails, we never lost that tail. It's always there in the embryo, and then you lose it as you develop. And then at last we have the structural homologies, which are similar structures in adult organisms. A good example is the bones in the arm, which you can find in all these different species. So in the human, you have the humerus in your upper arm. In your forearm, you have the radius and the ulna. Your wrist bones are the carpals, your hand bones are the metacarpals, and your finger bones are the phalanges. And you can find all of those bones in a horse, in a bird, in a bat, in a seal. So the horse, bat, and seal are all mammals, but the bird's not even a mammal. It's a bird. And birds have evolved from reptiles. So all these bones must have come at least from reptiles. And actually, if you go back to the transitional features slide, you can find these bones again. Well, maybe not all these hand bones quite, but you can definitely see the humerus, the radius, and the ulna. You can even see them in fish, and you can see, you know, what would have corresponded to some of these hand bones as well. It's just weird that we look so similar if we're not related. And the best evidence for evolution is the fact that you can actually watch it happen. In general, we think of it as being a very slow process that you can't actually see, but sometimes you can see it. So we actually are able to observe speciation events today. A species is defined as organisms that are able to breed with each other and produce viable offspring. So if you have a horse and a donkey and they breed together, that gives you a mule. A mule is actually sterile, it can't have offspring. So that's why we consider horses and donkeys to be a separate species. But actually some organisms that are able to breed together and produce fertile offspring are considered to be in different species if they actually don't breed together in practice. They have to actually breed together to be considered one species. If they're kind of separated so that one of them comes out during the day and the other comes out at night and they never meet each other, then they're different species because they don't actually interbreed. So some of the recent speciation events that we've had would be the big bird finch, which you can see right here. So as I had mentioned, they're continuing to study the finches on the Galapagos Islands. This is a new species of finch that has recently evolved on the Galapagos. It came about as a hybridization between two other species. So you have the G. fortis finch and the G. canerostris finch on the Galapagos Islands, and these two finches normally do not interbreed. That's why they're considered different species, but they are actually capable of breeding, apparently, because something freaky happened and they did breed. And then you got these hybrid parents, which bred together to create this new finch, the big bird finch, which now does not breed with either parent species, only breeds with other big bird finches. So that speciation event occurred over the span of 3 generations. You just needed 3 generations of hybrids to create that big bird finch species. Killer whales are actually speciating right now. That hasn't happened yet, but we're really pretty certain that it will happen sometime soon-ish. Of course, they kind of have a long lifespan, so they don't evolve very quickly. But you have all these different types of killer whales and the different oceans where they live. So, for instance, in the North Pacific, you have these 3 types of whales. The offshore whales that live far away from the shore, they eat this fish. The resident whales live closer to the shore, they eat this fish, and the transient whales just move around all the time and they eat seals, primarily. So these different whales live in different places in the ocean, and they also have different foods that they like to eat. And they also look a little bit different from each other, and as time passes, they're going to become more and more and more different, and eventually they may stop interbreeding with each other, at which point they would be considered different species. And then finally, we have a new maidenhair fern. I wasn't able to find a picture of it though, you guys. But this maidenhair fern was created in a non-destruction event during meiosis. So what that means is that when the chromosomes were all supposed to split and go to different sides of the cell, so you can have 2 new cells that have each a complete copy of the chromosomes didn't split, so they all went to one side of the cell. So you got like one cell that has no chromosomes and just died, and another cell that had like extra copies of everything. And if that was a a sperm cell or an egg cell in people or in mammals, that would not have been viable. That would have just died. But since it was in plants, they can just handle having extra chromosomes actually really well. So that created this new species of maidenhair fern, which has a completely different number of chromosomes from its parents, and does not breed with the parents, and is its own new species. That happened in a single generation. And finally, the most important aspect of evolution for us today is antimicrobial resistance, the development and evolution of antimicrobial resistance. So unfortunately, you can watch evolution happen in the hospital as these organisms that used to be killed by antibiotics gain resistance and are no longer killed by antibiotics but kill us instead. So actually, according to the World Health Organization, strains of bacteria that are resistant to antimicrobial drugs are the biggest health threat faced by our species today, and it's estimated that by the year 2050, more people will die every year of resistant infections than of cancer. So the way that develops is that you start with a population of bacteria. Most of them are susceptible to the antibiotic, which means that the antibiotic will kill them, but you have a couple, maybe even just one, that has a mutation that makes it resistant to the antibiotic. So the antibiotic doesn't kill it, or maybe it takes more of the antibiotic to kill it. And then you take the drugs, antibiotic drugs, all these susceptible bacteria start dying. So at the end of your course of antibiotics, you have just very, very few of those susceptible bacteria left over, maybe none left over, maybe they would all have died. But these resistant ones, they didn't die. They're not susceptible. The antibiotic does not hurt them, so they're still alive. And now all of their competition is gone. They used to be competing for resources with all these different bacteria. Now all those bacteria are dead, and there's nothing there but them left. And they start multiplying and multiplying and multiplying until they are the bacteria that are infecting you. Now you have this whole new population of bacteria, and almost all of them are resistant to the antibiotics. If you take antibiotics again, it isn't going to do anything. Here you can see how they test for antibiotic resistance. So they plate a lawn of bacteria on this petri dish. So you basically just take a swab get the bacteria on it and then smear it all across the whole dish, and then you wait for some days, you incubate it for some days so the bacteria grow and you get this mat, this is called a lawn of them all across the whole plate. Then you put these little discs on there. Each of these discs has been soaked with a different antibiotic drug. And if you wait, the antibiotic is going to kind of seep out into the petri dish around the disc, and it's going to start killing bacteria, and then you'll get these circles where you don't have any bacteria. The bigger the circle is, the more deadly the antibiotic is for the bacteria. They're not able to live close to the disc. They have to be far away to survive. So this bacteria here is susceptible to all the different antibiotics that were tested. This one, it only kills the bacteria a little bit, but it does kill them some. This one kills them a lot. They have to be very far from that disc to survive. On the other hand, if you have a strain of bacteria that is resistant to antibiotics, it'll be resistant to some, but probably not to all of them, probably, hopefully. So if you put those little discs on there, some of them will get the little clear zone around them, and some of them will not. This bacterial species is resistant to all of these antibiotics, all 5 of these, and it's almost all the way resistant to this 6th one here. It's not extremely susceptible to any of them. None of them are able to kill it extremely well. If you get sick with this, that's going to be very difficult to treat. Here you see a graph of the rise of these strains of antibiotic-resistant bacteria over time. The one that you may be familiar with that you might have heard of is MRSA. MRSA is methicillin-resistant Staphylococcus aureus. That was first discovered in the 1970s, and it was very rare at that time, and we've been tracking it through time. Every time someone has an infection of staph and they go into the hospital and you figure out what type of staph it is, if it's resistant to the antibiotics or not, then you can measure the incidence of resistance over time. And as you can see, it grows and grows and grows. This graph doesn't even go till like now, it just goes to like 2005 or something like that. And you can see by 2005, almost 70% of all strains of Staph aureus that were making people sick were resistant to methicillin. MRSA is now the dominant type of Staphylococcus aureus that you find. So since these strains are resistant to methicillin, that used to be the antibiotic that people used to treat staph infections, but now you can't use it anymore. So people started using vancomycin instead, a different antibiotic. But now you have strains of Staphylococcus aureus that are resistant to methicillin and to vancomycin, and that process is just going to continue over time. These are other strains of different bacteria that are also resistant to different antibiotics, and as you can see, all of them are becoming more common over time. This is going to continue. Now you have strains of bacteria that are resistant to many antibiotic drugs. And as you can see, this process doesn't take crazy long. It took from the first incidence of MRSA to 2005, when it was 70% of all staph. That was only 30 years. We're not developing new antibiotic drugs very quickly, you guys. It takes a long time and it's very expensive, and there's not a way to make good money off of it. So it can cost upwards of $1 billion to develop a new drug, and it can take over 10 years. It can take over a decade. And pharmaceutical companies don't want to invest all of that time and money if they're not going to make a lot of money afterwards. And we like to have antibiotics that are kind of cheap and affordable so that people can buy them. And so they don't see a way to make good money off of it. And if no one's going to get rich off of it, then no one's going to do it. So we have very few antibiotics that are being introduced to the market these days. We are not keeping up with these bacteria whatsoever. They're going to overwhelm us. And that's why more people will be dying of these antibiotic-resistant strains than of cancer by 2050. We're going to go basically back to the dark ages, where any little cut can get you sick and kill you, and there's nothing that the doctor can do. So if we start living in that world, then it's going to be extremely important to wash your hands and not touch your face. Those things will be the kind of your main protection against infection when we don't have drugs anymore that can kill infections. I'll conclude with going into extra detail with this particular bacteria, Mycobacterium tuberculosis, which causes the disease tuberculosis. So first I'll talk about that bacteria's life cycle, and then I'll talk about the resistance that we're seeing with it. So Mycobacterium tuberculosis is abbreviated MTb. MTb likes to live inside of macrophage cells. Those are a type of immune cell that you have. It's pretty much in all of your tissues, roaming around looking for bacteria to kill. But Mycobacterium tuberculosis doesn't get killed when it finds a macrophage. It likes to live inside of the macrophage. So you would breathe in this bacteria, the Mycobacterium, into your lungs, and you have macrophages living in the lungs. They're scanning for things that you would breathe in that would be dangerous for you. And so this macrophage is going to eat the bacteria. Usually when the macrophage eats a bacteria, it destroys it. But MTb is resistant to that. It's able to live inside of the macrophage without being destroyed. So now you have this macrophage that's infected with MTb. This macrophage is going to start releasing chemicals that attract other macrophages. It's basically saying, there's a problem and I need some help. And so other macrophages will come. They'll come from the blood, and they'll also get infected. So after that's been going on for a while, you're going to have a little cluster here of all these macrophages that have come to help the infected macrophage. But they can't help. They can't help. All they can do is get infected themselves. This is called a tubercle. It's also called a granuloma. When it starts, it looks like this. You have your infected macrophages in the middle. You have other macrophages around the edges that are becoming abnormal in their shape. You have other macrophages even beyond that are just waiting to get into the middle. And then you just have regular lung cells. As time passes, this tubercle is going to develop what's called a fibrous cuff. It's like kind of a shell of collagen that's around it, and all the macrophages that are infected are kind of trapped in there. So at this point, you have a fully developed, mature tubercle. It would look something like this in the lung, a little white ball. And at this point, your infection will be latent, which means that you're not really having symptoms, and this is kind of okay, actually. I mean, obviously it isn't the best to have this in there, but it's not the worst either. So what's not good about it is that this cuff is preventing your immune system from getting in there. Macrophages aren't really helping you, but you have other immune cells that could kill the bacteria, and they're not able to get inside of there because of this fibrous cuff. But the good thing is that the bacteria are trapped in there as well. They can't get out. They can't damage more parts of your lung or infect new macrophages. They're stuck in there. So as long as you have this mature tubercle here, that's a latent infection, you're not really going to have symptoms, but you will be infected. And this can go on for many years until the infection reactivates. What causes that to happen, we're not really sure what causes it, but eventually it does happen for a lot of people. It might take a really long time. It might not take that long, but it usually happens. These bacteria are now going to be released. The cuff that was around the outside of this tubercle is going to get degraded, and it ruptures, and all the bacteria will escape now. And they're going to go elsewhere in the lung. They're going to infect new macrophages and make new tubercles. And also you can cough and sneeze them out. You're going to be having symptoms now. So now you can infect someone else by coughing it out. Here you have, this is a histology of a tubercle in a mouse lung. And these are some of those mouse lungs. Well, this is a mouse lung also. So these are all the lungs of little mice that were infected with TB. And these are the granulomas, the tubercles. As you can see, these mice have a lot of them in their lungs. So that's multiple phases of latency, then reactivation, and latency again. But of course, these mice that have such huge tubercles in their lungs were having a very hard time breathing. That's a lot of lung tissue that's been destroyed at that point. If you look at the histology, which is the tissue, if you section the tissue and stain it and look at it under the microscope, this is what one of those tubercles looks like. If you zoom in really, really close in the microscope, you can see the little purple things in there. Those are the bacteria themselves inside of the macrophages. And so that's how MTb goes about its business. We used to have, well, we still have this drug rifampin, which used to be really good at treating tuberculosis. People who would take rifampin would have to be taking it for a very long time because most bacteria divide quickly, but MTb divides really slow. Actually, it takes a long time for it to multiply, and the rifampin basically attacks it when it's multiplying. So it takes a really long time, treating with rifampin every day, to actually clear the infection of MTb. And what started happening is that patients who were treated with rifampin, they would be taking it for months. They would get better, and they would seem like, they're better now, and their MTb, their tuberculosis infection has been cured. But then they would come back months later, they would come back to the hospital and they would have the symptoms again. And if you look at their lungs, they have the tuberculosis again. And you treat with rifampin again, but this time they're resistant to it. The bacteria they have is not susceptible to rifampin anymore. It is resistant. And so it doesn't help. The rifampin doesn't help them anymore. And they get sicker and sicker and then they die. And that is because of the evolution of rifampin-resistant Mycobacterium tuberculosis. So rifampin actually works by targeting the ribosome. Here you have the ribosome of MTb. Right here in the center is the active site, where you would actually have amino acids being linked together to make a protein. And here in red is where rifampin binds to the ribosome. So rifampin is supposed to come in, it's supposed to grab the ribosome right here, and then it changes the shape of the active site so that amino acids cannot get inside of there. And then the ribosome is basically broken, it doesn't work, and the mycobacterium is not able to make new proteins, and so it will die. Especially if it's trying to divide. If it's trying to divide, it needs to make double of everything it has, so it needs to make a lot of protein, and it can't do that, and so it dies. This is the sequence of amino acids that you find right here where rifampin is supposed to bind. So this is part of the part of the primary structure of the ribosome right here. In these little boxes, you have all of the mutations that confer resistance against rifampin. So if you have any of these are all point mutations. If you have any one of these mutations occurring in this gene for the ribosome, you're going to end up with Mycobacterium tuberculosis that is resistant to rifampin. So for instance, if you have a leucine and a serine here, that's susceptible to rifampin, that MTb is going to die. If you have methionine there or leucine there instead, now that Mycobacterium is resistant and it's going to live. These are a lot of different mutations that all confer resistance. So every time that you get a new rifampin-resistant strain, you have one of these mutations. Could be any one of them, just by random chance. So you start out with your Mycobacterium tuberculosis infection. Most of them don't have a mutation. They're susceptible to the rifampin drug, but you do have just maybe one, maybe even two that have one of these mutations in their gene for the ribosome, and that makes them resistant. So then you treat with rifampin, and all these susceptible MTBs start dying over the months as the months pass, but the ones that are resistant, they don't die, they still live. And now they don't have any competition. All the bacteria they were competing with for resources, for food, they're not there anymore. They're alone. And so now they thrive and they take over the population, and you come back and you have tuberculosis symptoms again, and now you're sick with these rifampin resistant bacteria, and now rifampin does not help you anymore. So this is, real serious. This is really, really serious. If evolution wasn't real, it wouldn't be happening, but evolution is real and it is happening. And, you really need to wash your hands, especially if you're going to be working in the hospital or the clinic. That's the #1 place to find bacteria that is resistant to antibiotics. It's the #1 place to find it. You don't want to get sick with it. You don't want to bring it home to your family. So that means you got to wash your hands 20 seconds with the hot water with the soap, or with hand sanitizer once hand sanitizer becomes available again. And, just be really careful about your hand hygiene, you guys.